Good morning, everybody. Thank you very much for joining today's webinar. Today's webinar is on CombiRIDGE, which is a generative neural network powered GPU docking engine for ultra-large library virtual screening. My name is Andrew Orry, and if you have any questions after the webinar, please feel free to email me. The software that we'll be showing today, CombiRIDGE, was developed by Max Totroff, our Chief Technology Officer, Ruben Abagan, a company founder, and Eugene Rausch, the principal developer, who is a panelist on this webinar as well, to answer your questions. In this webinar, you should be able to use the Q&A button in the Zoom to ask questions. I'll try to answer them as we go along, but most likely we'll cover the questions at the end of the webinar. I think the chat itself is disabled, so you can only use the Q&A option. So in this webinar, we're going to explain what CombiRIDGE is, the general workflow for CombiRIDGE, applications of the method, some case studies, what hardware and software you need, what databases you can screen, and we'll also show a live demo of CombiRIDGE. So CombiRIDGE is a new and innovative solution for high throughput ligand docking and virtual screening. It's fully GPU accelerated and integrates three parts of MuleSoft's key technology. So the first is the one in the middle, Ginger. So Ginger is a generative neural network method for producing accurate and diverse conformer ensembles. Ridge, as far as we know, is the fastest docking method currently on the market. It's a high-performance GPU docking engine capable of processing ultra-large libraries at unprecedented speed. And then we have a scoring method called RTCNN, developed by MuleSoft. It is an advanced graph neural network scoring function that improves the accuracy and reliability of your hits prioritization. So this combined approach of these three methods enables CombiRIDGE to run. And it allows you to optimize specific R groups within a scaffold, but also to efficiently explore ultra-large chemical spaces and ultra-large like combinatorial libraries. And this sort of makes previously intractable screening campaigns both feasible and practical. I just have this representation in ICM just to show you what the overall steps are. So you would take a crystal structure like this. You may have it co-crystallized, but if not, you could run ICM Pocket Finder and find different pockets within the structure. You prioritize one particular pocket, like this one, for example, and then you take ridge and you dock these anchor molecules to the pocket. So you dock many millions of these. You evaluate the interactions, identify hydrogen bonding patterns or any kind of good interaction, and this will be reflected in the scoring that we have. And then you select the best scoring anchors, and then Ginger generatively generates the rest of the ligand, the full ligand, in the pocket uh, on the fly, and enumerates the full ligand based on the initial scaffold shown here. And then you have many. So you have your initial fragment, and then you have many uh, other ligands that have been scored, and you can use those to rank and then prioritize for experimental testing. This workflow just summarizes what we just saw. So the first step is it's an anchor-driven approach, CombiRIDGE. So you need to have an anchor conformational database. So you have your anchors, the fragments. You use Ginger to convert those into conformers, and then you have the conformational database. So MuleSoft has already generated these databases for you to use. So really, you just need to jump to step two. And step two is the docking of the anchors using Ridge. And once you have the top scoring anchors, you can then use Ginger to generate the full 3D conformers directly within the binding site, which we just saw. This enables very sort of rapid combinatorial expansion of the pre-docked anchor structures into the complete compounds. And so effectively, you are sampling the chemical space around each anchor. Next is the pose refinement, and it's carried out using GPU accelerated grid and MMF molecular force field energy minimization, which improves the accuracy and consistency um, of the predicted binding poses. And then finally, you can score those dot poses using either MuleSoft's physics-based score or MuleSoft's neural network score. And we'll cover both of those in a bit. So as a summary of why you might want to use CombiRIDGE. So CombiRIDGE goes beyond the traditional chemical catalogs. It opens up new regions of chemical space where compounds are enumerated and conformers are generated on the fly. This allows us to screen ultra-large databases far more efficiently, delivering sort of dramatic acceleration compared to more conventional methods. Importantly, when you're screening these large databases, is the concept of synthetic feasibility. So this is built directly into the process, ensuring that the proposed compounds that you get hits for are practical candidates for follow-up chemistry. The approach also supports efficient exploration of chemical diversity. And we're seeing this a lot in our contract research projects that we run here, that you identify new chemistry. It's possible for many reasons, but one reason that maybe is that the initial fragment that you're docking, the anchor, can maybe identify sub-pockets within the binding site which may have been overlooked when you're docking, the regular full-sized ligand. So this first step of docking this anchor could have that effect. We've also anecdotally run CombiRIDGE on quite a few projects for pharmaceutical companies and biotech companies in our own contract research projects here. And we see great success with typical hit rates probably between 5 and 10%, but substantially higher for some projects depending on the target. So if you want to use CombiRIDGE, then you need some specific hardware. So for running CombiRIDGE and our other GPU accelerator tools, we recommend sort of the following specifications. So you would need an NVIDIA GPU, RTX 3090, or preferably better if you have access to maybe 4090 or something like that. Then that would be good. Um, you need at least like 24 gigabytes of RAM, but ideally a bit more. CPU you need a minimum of 8 cores, although 16 cores is recommended for best performance. The disk requirements will vary depending on the size of the libraries you intend to screen. But for example, just for reference, a multi-file, a 1 gigabyte of disk space could save 1 billion compounds. And so, for example, for the enamine CombiRIDGE anchor library, which contains 48 billion compounds. This occupies about 55 gigabytes of space. And finally, it's important to also install CUDA. The the machine shown here is one we use quite regularly here at Molsoft. It's just a regular gaming GPU with a reasonably priced GPU, and you can run CombiRIDGE on a machine like this very efficiently. Obviously, if you want to uh, have more performance, you can run in parallel on multiple GPUs, depending on what you have available. So if you have uh, a data center, we support both Ampere and Ada uh, NVIDIA architecture. So yes, so most of the benchmarks have been done on RTX 4090 on a similar gaming machine to this. So for the software requirements, uh, you would need ICM Pro version 3.94A, which is the release, and then you need a license for ICM Pro, which is our main desktop modeling software, and VLS, and a Ginger license for the expansion of the ligand of the fragments in the pocket using CombiRIDGE, and then Ridge for the docking. We use FlexLM or LMX license software, and CombiRIDGE runs um, on Linux. So you can use the ICM Pro software initially to set up your CombiRIDGE docking project, and there's also a variety of other tools embedded in ICM Pro, such as bioinformatics tools, sequence alignments, propagating sequence to structure, molecular dynamics, sometimes useful for maybe post refinement of your CombiRIDGE hit list. You can refine the ligand using MD. We're not covering that today, but that's another option. There's also things like the 3D ligand editor, which we developed with medicinal chemists at Novartis, where you can improve once you have a hit from CombiRIDGE, you can then improve that interactively in a 3D environment where you can make modifications and see the effect of those modifications on your ligand. Okay, so the available spaces that we have are pre-prepared. These are prepared by Molsoft. Uh, we have ChemSpace, and we're very grateful to be able to work with these great companies such as ChemSpace, Enamine, and Wuxi, who have kindly provided um, the data, the spaces. So we have the ChemSpace has 140 billion chemicals in it and 22 million anchors. So you dock the anchors and then you use Ginger to generate the rest of the space. Enamine's 48 billion and Wushi's 15 billion with 35 million Wuxi Galaxy database. We're also working with Crystal Pi, a very innovative company. We hopefully will have that database available soon. And we've been working for a while with e-molecules, and they have a particularly larger space of around 7 trillion, I think. But we're hoping to have these coming on board soon. But these databases are available for you to download. Okay, so what you download a set of chemical reactions from different vendors um, into a compact form. And these contain the Markuch core and the R groups 1 and 2, and they're saved in ICB format. You also need to download the anchor libraries. So these you have the anchor, which we saw, and then you have these caps where the R group 1 and R group 2 are placed. Um, And these are saved in multi format. So you need to download the software, you need to download the reactions from the vendors and the pre-generated conformers of the cores. So we support both this is a typical, this is from Enamine. So they have like a two-component reaction and also a three-component reaction. But in each iteration, one of the capped R groups is swapped out for the full set of corresponding synthons. So, and then for a simple two-component reaction with just two R groups, a single iteration is enough to complete the molecule and produce a fully uh, full structure for ginger. We pre-process three-component reactions into two components by enumerating just one of the R groups. So these are the two files you need to download for each of the vendors. We provide those. We provide the URL to download that to you. Just to comment on the multi format. So for ultra large virtual screening, containing obviously billions and trillions of molecules, fast and efficient storage file format is critical to support these workflows. So Microsoft has developed an innovative, highly compressed file format to meet this challenge. This format uses frequency-based adaptive encoding in internal coordinates, which drastically reduces the file size. So in data compression, sort of compression, the adaptive encoding technique is where frequently occurring data patterns are encoded using fewer bits, while the less frequent patterns use more bits. And it's extremely compact. So each molecule's conformation stack takes about 400 bytes per conformation. So these may have like 30 to 40 conformations per molecule. It's approximately 15 times more efficient than storing data in like regular Cartesian XYZ format. And the file extension is called .multi, and it can also be used with other algorithms inside MuleSoft, such as RIDGE, which obviously we're using now in this demo, and GigaStream. Okay, so we have our databases. We downloaded them. The blue. We have the reactions. We have the conformers of the anchors. So the next step is to dock using Ridge docking. So what is Ridge? It's a state-of-the-art docking engine designed to fully sort of leverage the GPU power, but it also efficiently utilizes the CPU resources on your machine as well. So it's kind of like a perfectly sort of balanced computational load. Ridge is fully compatible with your regular ICM docking projects. So if you're using regular ICM VLS, you can just take that project and use it out of the box. As we know, it utilizes pre-generated conformers for efficient performance. And then the docking engine operates in three distinct blocks. So we have the CPU block here, which manages the conformer databases and assigns charges. And then in the central part, we have this GPU block, which mainly handles the pose mining. And then there's a final step, which has the RTCNN scoring and the full atom physics-based scoring for enhanced accuracy. So uh, our regular docking, ICM docking that's been used widely over the years, the ligand is fully flexible. So the ligand is sampling the pocket as you're docking. It's been very accurate over the years and it's been applied to many docking drug design projects. And by using pre-generated conformers, we don't see too much loss of accuracy between pre-generated conformers versus fully flexible ligand. Maybe possibly about 10% loss. On an RTX 4090, you can screen approximately 400 compounds per second per GPU, which means that you can screen 10 million compounds very efficiently on a reasonably priced GPU. But by using Ridge with the CombiRIDGE method, then you can speed this up considerably. So the next step, once you've docked your anchors, you rank those anchors, and then you choose the anchor fragments that you want to enumerate. And the enumeration is done by Ginger. And this was published last year by Eugene Rausch and Max Totroff and Ruben Abagan. And I can send you that paper if you're interested in reading more about it. But basically, Ginger is graph internal coordinate neural network conformer generator with energy refinement. It's a cutting edge tool that uses state-of-the-art generative neural network-based algorithms, and it efficiently explores the conformational space of molecules, delivering diverse and low energy conformers. The conformers undergo fast gradient local force field energy minimization, which ensures accuracy. So the other features are that the GPU acceleration. So a library of 10 million compounds can be processed in just one day efficiently. The Ginger's generative algorithm provides accuracy comparable to leading industry rivals, and we have like maybe 40% smaller ensembles. There are also a variety of different customization options. You can generate conformer libraries of varying density and size to match your specific needs. Obviously, this scalability is the key strength here. It handles very large molecular data sets, even over 1 billion, obviously billions of compounds. It's pretty robust, and we're finding that generally it processes approximately like more than 99% of the compounds in your typical vendor databases. So once you need to score the anchors, and then once the Ginger has enumerated the full ligand in the pocket, you also need to score those as well. So one scoring method we have is called RTCNN. It's extremely fast, and it differs from our regular docking score in that it does not use molecular mechanics or physical energy terms. So we have one physics-based score, which I'll talk about in a minute, and this neural network score as well. So the RTCNN is trained to distinguish native-like complexes from decoys based on the geometries of the putative complexes in the PDB. So all favorable or unfavorable interactions are learnt from the training examples rather than any predefined input terms. A key distinction between the RTCNN score and the regular physics-based score is that the RTCNN score has a lower penalty for minor ligand interaction clashes due to, for example, induced fit, that kind of thing. A lower RTCNN score indicates better prediction for the ligand receptor interactions. So the other score we have is the physics-based score. So the scoring function considers various contributions, such as, for example, van der Waals interactions, the repulsive attractive forces between atoms, electrostatics as well, the interactions between charged atoms, hydrogen bonding, contributions from donor acceptor pairs. We have a desolvation term as well, which gives a cost for removing the water molecules from the binding site, and an entropy term for ligand flexibility, accounting for loss of degrees of freedom upon binding. It's also been recently, well, in the last few years, it's been updated with big data derived terms for ligand strain. So you'll get a penalty if your ligand is particularly strained. So this slide shows the workflow for combinatorial docking. This was done about a year ago when the real space was 28 billion compounds. It's now, I think, 48 or something like that. So the process begins, as we know, with approximately, well, for this 28 billion real space, there are 18 million anchors. So we pre-processed that using Ginger into the conformers. And just to give you an idea of the time for that, at a throughput of about 7,500 conformers per minute, this pre-processing step takes approximately 40 hours. But this only needs to be done once. And these are available for download from also. Next, the anchor conformers are docked using Ridge, and obviously the anchors are relatively small molecules with fewer rotatable bonds. Uh, Makes it ideal for efficient docking. And then the low energy poses are selected with an emphasis on the expandable pocket space around the attachment points. So you don't want to be you don't want your attachment points or the caps to be buried. So you want to have space to build out. And the GPU docking performance is extremely fast for this. You'll see approximately like 3,500 anchors per minute. And so this full database was docked in about 90 hours. This is just on a single desktop GPU gaming machine. And then finally, the top ranked anchors are expanded into the full compounds, and these are docked and scored like in situ using a combination of Ginger and Grid molecular force field minimization. And then, for example, the top 1,000 anchors then can be expanded and fully evaluated. And this took about 6 hours. So this gives you a rough idea of how long it's going to take you to screen just using a regular, uh, just a RTX 4090, I think it was done on, and obviously it can be parallelized as well. So we're going to start with an example, and we move into the live demonstration. Just need to. I don't want to dwell too much on this because we have other webinars that explain how to set up the docking. But basically, you read in the PDB, you convert it from PDB to ICM objects. So you're adding hydrogens and fixing protonation states and orientations of glutamines, asparagines, where there may be ambiguity from the X-ray structure. You remove the ligand from the pocket. You go to the docking menu, new project. There's some preferences, and then you can dock with the Ridge, dock using Ridge. The setup can also be fully automated using a script called dock batch. And if you look back on our YouTube channel, you'll find some examples of this. So I just want to go to ICM, just quickly show those steps. So we have a direct link to the PDB. You go to PDB search here. The example is a 3FSK, which is a MAPK kinase. There's an inhibitor bound. I'm going to go quite quickly through these steps, but as I mentioned, there are other webinars on this. So he's going to convert it to an ICM object. We could run ICM Pocket Finder, but we know the ligand is here, so we're going to use that pocket to dock to. So it's going to move the ligand out of the pocket, move from object, so there's space to dock. But conceptually in ICM, it looks like the ligand's in the pocket, but in reality, they're two different objects, so they don't see each other. So this is not there. And then we can go to the docking menu and choose new project. and go. I'm just going to call it mydoc underscore 3fsk. And then we have the receptor object and the um, we can define the site. We can select the ligand and then define the site around selected ligand. Here, with the green, we have the green crosses, and go OK. And then this is a probe. Go is the size of the box is where the docking is located. So with CombiRIDGE, you may need to be careful because if you want to expand, in this example, we probably don't want to expand too much here, but if there's a sub pocket you want to target, you need to make sure that the purple box covers that region. And just press go and it will. So the structures are so now it's ready for docking. So now everything is ready for CombiRIDGE. Uh, it will generate a number of different files. So we have in this example I called the project doc1, but in our example it's called mydoc underscore fs3k. These are the files you need for the CombiRIDGE. So we have the docking maps. A template can be used, receptor objects. You can also use multiple receptor conformation as well, which we'll talk about in a minute. But these are the files you need to run CombiRIDGE. You can also add some options. So you can include a template if you want to guide your fragments, your anchors. You can place like a chemical at this point, a chemical fragment, and say I want all the anchors to try and dock into this region and match this substructure. Or you can use a 3D pharmacophore template. So this is a method called atomic property fields. Again, there's a whole webinar on atomic property fields. But you trying to match. So when you dock the anchor, rather than matching substructure, it's going to match the pharmacophoric property. So you can say I want a hydrogen bond acceptor here, hydrogen bond donor here, and a lipophilic region to match here, and that will guide your anchors to the pocket to a particular site in the pocket if you want to do that. You don't have to use these options. And the other option is to use when you're docking the anchors is to use multiple receptor conformations. So we have a method in ICM called 4D docking. 4D docking is an ensemble-based approach to docking. So you can use one or more. In 4D docking, you use more than one. So you use two or more crystal structures or models that represent the flexibility of the binding pocket. So you could also use snapshots from MD. And so you dock the anchors not to just one receptor, but to multiple receptors in one go. So this is over many years, most of us we've been working on induced fit docking, and this is a very efficient way of doing multiple receptor docking. Okay, so this is the setup. So practically to run it in ICM, you can actually run Ridge in the graphical user interface, but I think you're better for the webinars. We look at the actual commands. So you need to first call ICM, and then you call the Ridge program, and then you give the project file name. So that in our case, that would be mydoc whatever the PDB code was. And then you have the output in SDF format. There are some options for Ridge. We can look at those later. And then you have the input. So these are the mol-t files. So these are the 3D conformers um, of from each of the vendors. And you can also screen multiple vendors in one go. So you could have Freedom Space, Real, and Wuxi screened in one go. In this example, we will use just someone move them. We will just screen a random set. So it's going to go to try and dock these 10,000 in one go. We have I'm going to just choose a random set of 10,000 anchors from the enemy space. So paste here. And it's running. I'm going to talk about what I just pasted in here. So it's running. While it's running these 10,000, I will go back to the screen. So what I ran was called ICM, called Ridge. The docking project is mydoc_3fsk, not doc1. The input was the multi file, which I have saved in this. You have to give the path to where that multi file is saved. There are some options that you can use. So -C is it performs the Cartesian optimization for the best pose. Minus uppercase S is the rescoring with the physics-based score. And then there are some other options that it's going to MN hits and random selection. So this is some more options in the ridge in the ridge thing. So it's got through 6,500 already. It's going to get there. It should be a minute or so, couple more minutes to finish. So once that's done, our output file will be an SDF file, and we can and basically that SDF file will be the anchors all docked and scored, and you can see the interactions. Okay, it's coming to an end. So it's finished docking those 10,000 anchors. And then we need to go back, we need to go to the results file. So to do that, let me just open ICM. I have it saved rather than transferring the file I opened. I have it here. Okay, so the results of the anchor docking will obviously be a hit list here with your anchor. You can sort the column, you get the smaller ones. We have a scoring. So this is a combined score of the RTCNN score, the physics-based score, and also some penalty in case the fragment is not optimally docked in order to expand upon. So we can sort by score here. And another few things we could do. So we basically we want to leave want to maybe remove some of the poorer scoring fragments. We don't really want to enumerate those using Ginger. So what we could do is we could plot, for example, we could plot the score, which is the physics-based score, against RTCNN score. We could color by strain, and sorry, we could have the point size based on the number of atoms, the size of the ligand, and then the strain we can color by strain. So this will give us a plot. Yeah, the points are very large. I might just for clarity, I'm going to put the size points just back to a regular fixed size of 6. It's a little bit easier to see. So, for example, we could use we have a column called score combined, but you could also cherry pick your own fragments that you want to um, build, build from in the CombiRIDGE. So you'd ideally choose those that have low physics-based score and those that have low we have this very good scoring one here, RTCN scoring one here, which is our top score. So you can select here. If, for example, we rank by score, I just want to get a slightly smaller database so I can show you a slightly smaller hit list so I can show you some more analysis tools that you could do. So I'm going to keep the top 150 um, compounds. So I'm just going to delete these rows. So I should have also said you can scroll through the table and see the ligand bound to the receptor here. You can use the cursor keys to just scroll through these and see how the anchor is positioned. Um, this is a kinase, so maybe we're interested in only those anchors that make a hydrogen bond with this region in the hinge. So we can select that atom, go to Tools, and then we can say um, hydrogen bond to selection and give it a number. And there's a calculate property. So this will go through all 150, and we should see an extra column at the end with the hydrogen bond energy. So this one should have, yeah, you see it makes a hydrogen bond with that hydrogen. If it's 0, you shouldn't see any. Yeah, this fragment is obviously away from the hinge. So we may want to, if we're not interested in those kind of poses, we may want to remove those before moving on to the next step. Another thing you could do is in the tools panel, there's a variety of tools. So you could also compare you could compare the fragment with known chemistry. So you would load a co-crystal structure and look at the pharmacophore RMSD, or you could look at how well it matches the 3D pharmacophoric properties of known chemistry. If we look at the interaction of fingerprints, we can make a column called fingerprints. And this gives you this column here, which we can cluster. So if we cluster that using the clustering tools here, just go OK. We get a tree. So this could help also removing some fragments that you don't want. So these two are likely to be in the same position. You see they're making hydrogen bonds with the hinge. These are, if we go to a different, further down the tree, we can probably see these two have different poses. They should be pretty similar interactions. And this one's a little bit of an outlier. It's on its own, a singleton. You could remove that. This tree is fully interactive with the table. These ones are making nice interactions going towards the allosteric site here of the kinase. So once you've fully analyzed the anchor hit list, you can move on to the next step. So the next step is to run Ginger. So well, Ginger, but inside the CombiRIDGE script. So again, practically speaking, to run it on the command line, you call ICM, you then call this script called CombiRIDGE, your project name, and then the input fragment database. So this is this SD file that I just read in. And then you need to define which space you want to search. So you could search, you could download the reactions in ICB format, which is ICM's format, and then uh, um, or so you download these from for each of the vendors, and you can search those. We'll probably answer questions regarding all the flags and stuff maybe at the end, but it's all documented so you can read it. So you can apply formal charges to the ligand, um, calculate ICM scores, and so this example, we enumerate the first 10 um, anchors from the ridge run, and each anchor will produce about five, well, about 1,000 final compounds on average. So you run this on your you need to give the if you need to give the path to the SD file that with the anchors docs, and then you will see a um, sorry, you will get a similar thing as we saw before with a hit list, but now we have the full um, ligand bound. You can screen through them. So the top scoring one from the anchors, I didn't show it very well, but I will show now. Um, yeah, so the top scoring anchor, you can see it is we sort by score was this one, which had the lowest combined score. So there's this like halo group on this thiophene ring and the amide here. So if we go back to our results, we should be able to find we have that same initial scaffold, but we can look for the other ones with that same um, with that same with the same scaffold. So we can use the tools in the column. So we have a variety of information that's telling you which database we screened, and then we have the oh this is so wrong. So when it's docked, sorry, we get this you just sorry, I just need to get the results up from the screen from the CombiRIDGE itself. Um, file, recent files. Ooh. So when CombiRIDGE run, you get this. Sorry, I was showing the wrong file. I was showing the anchors. So you can see that we have um, the top hit is has this same anchor as which we saw before, but obviously with the enumerated differences. So we can see all of those by just looking at this reaction ID. So all of these with the same ID have the same anchor, basically. So if we filter the table by this option, go to filter, um, and then filter the column reaction ID. Um, contains. Okay, so we get this little symbol here, which means that this column is filtered. So now all of these should have the same anchor. So if we just zoom in on the ligand, this is that group here and the amide. So now if we display these, we can see how this group is growing into this region. See the anchor is the same, and then select all. Select a few of them. You can just display. You can display them like this. And then you would obviously rank by score again. You can also do the similar filtering Just gonna change the display a bit. Uh, you could also do the filtering as before with the anchors, but this time with the full ligands. You can cluster and um, and then choose for experimental testing maybe a diverse set of anchors and full molecules or whatever you would rank basically on the score combined, or you could choose the RTCNN score itself. So just as an example, this is a case study using a very uh, tricky molecule. It's unusually large, 867 molecular weight. It's called venetoclax. Pretty familiar with it. It's the first in class inhibitor of BCL2. Uh, so BCL2 is an important drug target. It's a pro-survival protein, regulates apoptosis. And so this is a challenging example for CombiRIDGE. So basically, the compound in the study they took 1 million anchors and docked those anchors. These anchors were taken from the real, and with a hypothetical venetoclax synthon, and you can see it scored very well. And then after CombiRIDGE enumeration with ginger and pose minimization, venetoclax was successfully redocked, within zero point 0.7 angstrom. So you have the anchor, and then this extremely large molecule was built using ginger and CombiRIDGE on top of the original anchor here and docked extremely well compared to the PDB. So I just want to finish up with letting you know that we do have other ultra large library screening tools available. Many of you already use ICM Pro VLS, which is our CPU version of screening. There's been a long history of success stories using VLS. It's about approximately 1 to 2 chemicals per second per CPU. And a couple of years ago, Seva Katrich's lab at USC developed a method called ICMV Synthes, which is a great tool for ultra large library screening as well. It docks fragments and then enumerates those fragments, and then you dock the full ligand to the pocket. So these tools are available in ICM. And then the GPU tools, we talked about Ridge already, we talked about CombiRIDGE. But we also have GigaScreen, which is available. It combines machine learning and deep learning tools. So it combines both Ridge, which we've already discussed, RTCNN scoring, and also a GCNN chemical model. So it's basically training. And one of the advantages of GigaSearch is that you can obviously screen very quickly in one day on one machine. We also have a tool called RIDE. It runs on CPU or on GPU. It's extremely fast. It is a ligand-based screening method for ultra-large libraries. We have pre-prepared libraries from a variety of vendors that you can use. So it's a good tool for if you have maybe a lead compounds, but you don't have a crystal structure, you can identify scaffold hop to new chemistry using the atomic property fields. And rather than matching substructure, you're matching the 3D pharmacophoric properties. And it's about 1.5 million chemicals per second per GPU. And one of the reasons you can also use this for structure-based screening as well. So you can combine the score of how well your ligand matches these fields as well as the structure-based score. as well. So these tools are, I just wanted to point out, these are all available. And so just as a summary of so vSynthesis uses ICM VLS, which is our CPU-based approach. And in this example, we do full redocking of the vSynthesis, they call the anchors MELs. Um, and we used CombiRIDGE. And for both methods, we compared the top 250,000 compounds, first by VLS score and then by combined score. We also looked at the median score values for the top 10K, 5K, and 1K hits. And the results are pretty clear that the combined score for CombiRidge is consistently better across all targets. So this is a vSynthesis project that we did last year for some pharmaceutical companies, and we compared the results using CombiRidge basically. So for but for VLS scoring alone, the vSynthesis outperforms CombiRidge primarily because the CombiRidge protocols are finely tuned really to the RT-CNN scoring. So yeah, so both protocols are available. CombiRidge uh, is available as well. So we covered lots of things today. So if you want to go back and see how to set up your docking project, there's YouTube videos online. There's also videos on how to incorporate induced fit. So the 4D docking is there. We have some manuals and also a knowledge base where you can ask questions if you want to, or you can direct questions directly to us by email. So yeah, feel free to email after the webinar, and I'll try and answer some questions that came in by text. I think some of them have already been answered by Eugene. Thank you, Eugene. So one of the questions was, what is the dash anchor flag doing in the underscore ridge run? So this flag removes anchors where the cap is buried in the pocket. So there's no space to grow. I don't know if you're able to see Eugene's answers or not. So, what was it? Um, another question was during enumeration, can simple filters such as molecular weight, hydrogen bond acceptors, donors, limits, or structural alerts be applied, such as panes or bad groups? And Eugene answered, yeah, there's an option to do the filtering during the enumeration step, and it uses the filters that you set in your docking project. So we didn't quite cover that because we didn't have much time, but in the docking preferences, you can set different filters here as well. There was a question about whether the anchor could be covalently bound. I believe Eugene, Max, and Ruben are currently thinking about this, and it could be included in the protocol. Um, I had a question about the induced fit changes. So yeah, the probably best. So the 4D docking can be used in the ridge step, docking the fragments. Um, but I would guide you to the webinar, or there's a paper describing the 4D method that we use. It's a question for you, actually. There's a question whether the selecting the fragment poses, is that automated? I guess in a way it can be, right, Eugene? I don't know if you can open your.
Yes, so basically, if you just use score as a main and as a primary filtering part, yes, you can just take your hits directly from the reach step and pass it to the CombiRIDGE step. If you want to do more fine filtering, as Andy already showed, you can like do some clustering. You can apply some additional stuff. By default, it will use combined score and will take top how many top fragments you want or how much enumeration space you want to explore. So it's easier basically to think in terms of how much molecules, enumerated molecules you want to finally dock in the last step. And there is a parameter in a CombiRIDGE where you say, okay, I want to dock 50 million molecules. In that case, it will just start from the top ranking anchor and start enumerating until you reach that amount, and your final hit list will contain top number of hits which you requested. So that's basically how it works.
Thank you, Eugene. There was another question actually, you may be able to help with. So for conformer strain, is that calculated after a group is grown onto the anchor, or is there a step there in Ginger, I guess, or
Well, I think Ginger must have this strain option. It should be as a separate column in the hit list as far as I remember. But if not, it's easy to add. But we have that strain because we have this three strain component, which also based on neural network on statistical torsion profile. It's another paper, and yes, that strain can be used for further filtering. It should be as a single column, or there must be some separate option in the CombiRIDGE protocol which will add this column into the final hit list.
thank you. The other question about the anchor library. So the anchor library you can download directly from Molsoft because it's fully prepared. So it has all the anchors, the conformers of the anchors already prepared. So we can send you the download link for that. Okay. And we're kind of up to the time. So I think some of the other questions we will get back to you via email. I just want to thank you very much for your joining the webinar and for your support of the software. And please feel free to get in contact with us if you have any questions. Thank you very much.